So it's my pleasure to introduce you the next speaker in this session is Dr. Luc Buée from the University of Lille. Dr. Buée, he did his PhD at the Mount Sinai Medical Center, and he's now director of a research center at the University of Lille Hospital. He has a long-standing interest in tau biology or tau aggregation and cellular functions, as well as in the development of cellular and animal models of disease. And he's going to talk about tau experimental models and therapeutic strategies. So, Dr. Buée, thank you for being here, and welcome to Alicante. Please.
Buenos días. That's the only word I can say in Spanish. So, first, I would like to thank the organizer and especially Jesus and Miguel for letting me giving you a few insights about tau. And as Eckhart told you yesterday, I think it's very difficult to talk about tau in Spain because, I mean, you have tau specialists. In fact, for a long time, tau was almost only studied in Europe before tau mutation and so on. So it's a kind of European tradition to work on tau. So I'm coming from a city in the north of France. It looks like a Spanish city, but it's really a French city, but it was a Spanish part somewhere in history. So finally, we're not so far. I changed a little my talk because, of course, we try to overlap the different talks on tau. So I must apologize because I think I have too many slides. So I will have to speed up on some of them. So the idea is really to, oops. Just trying to see if it works. Yes. To present my team first, I mean, I have a big team. It's about 40 to 50 people working in my lab. And they are all working on tau, either on tau genomics, tau splicing, tau protein, tau pathology. And we are part of a national consortium on Alzheimer's disease with people involved in genetics like Philippe Amouyel, or on amyloid like Frédéric Schekler, and also clinician neurologists like Florence Pasquier. And we have many collaboration in both France, Europe, and so on. So not all of them are here. So on today's talk, I will mostly focus on tau biology and mostly on non-microtubule tau functions. A few words on tau pathology, but not really related to Alzheimer's disease, but mostly related to other neurodegenerative disorders with aggregation of these tau proteins. And then I will finish on a few words on tau experimental models and therapeutic strategy we can have on tau, and especially a few words on tau secretion and tau spreading if I have enough time. Okay, as many people told you yesterday, we have tau aggregation in Alzheimer's disease, and you can see here that you have 6 tau isoforms in the human brains, and the 6 tau isoforms aggregate into filament to form neurofibrillary degeneration. This neurofibrillary degeneration starts in the hippocampal formation, then will go to the temporal lobe, then to polymodal association area, unimodal association area, and then you will have the full entire cerebral cortex with tau pathology and neurofibrillary degeneration. A few words on tau. You know that tau is a microtubule-associated protein. I'm not going to develop that it's under the regulation of phosphorylation, so you have a balance between kinases and phosphatases. But you know also that tau is not only linked to microtubule regulation, it has also other functions, and I will focus on a few of them where we are interested in the lab. Yesterday we had a question about tau in the nucleus, so I say maybe that can be interesting to talk about tau in the nucleus. We found that tau binds to DNA and is involved in DNA protection. It's also demonstrated by NMR spectroscopy showing interaction of tau with DNA. But tau not only binds to DNA, but also to any nucleic acid. And it may protect this nucleic acid from oxidative stress, for instance. So you can imagine that in neurodegenerative disorders, of course, you have a huge amount of oxidative stress, and without tau, you will have damage to nucleic acid. So that's what we have shown in these articles. What we have shown also, we and others, is that tau is also involved in chromatin remodeling. And especially we have shown that loss of tau affects the structure of the transcription and repair of neuronal pericentromatic heterochromatin. So it means that tau is a key player also in the nucleus, having some role in the chromatin, transcription effects, and also protection. If you think about tau, we did some transcriptomic studies in tau knockout mice, for instance, and we show that there is a huge variation in the expression of genes involved in DNA repair, for instance. A few words on tau and metabolisms. So for the lab, there is a joke about me saying I have a tau loss of function. Why? Because when you look at tau knockout mice, in fact, they are obese. And if you look at, and if you look at a model overexpressing tau, they are skinny. So it means that there is a balance between a loss of function where you get obese mice and an overexpression of tau where you get skinny mice. And recently we show that in fact tau was involved in the insulin signaling in neuron. And so we think that tau is also involved in this transduction signal. So you can imagine, for instance, that yesterday we heard about diabetes is a risk factor for Alzheimer's disease, but you can also imagine that a loss of function may trigger diabetes. So maybe a loss of tau function may be related to, may be instrumental in diabetes. If you look also to this tau loss of function, there is also a huge debate about decreasing tau because this is better for amyloid toxicity. If you decrease tau, you also change synaptic plasticity. So it means also that tau is involved in synaptic function. Finally, a few words on extracellular tau. Extracellular tau is used for, if some of you are neurologists or medical biologists, it's used for Alzheimer's disease diagnosis because you have an increase in tau concentration in CSF, and we know that there are extracellular tau. At the beginning, we thought it was related to neuronal death, and you get a release of tau. But in fact, more and more people think that there is a normal secretion of tau by neurons. We don't know exactly how it works. It's not unusual. If you think about FGF2 or interleukin-1 beta, you think, okay, that's secreted protein. But they are like tau. They are cytosolic secreted protein. So it means they're not going through the reticulum, the Golgi, and get secreted. They are, for instance, for interleukin-1 beta, there is cleavage in the carboxy terminal part of the protein, which allows the secretion. For FGF2, there is an oligomerization of the protein, which allows for the secretion. So you see, it's different cell biology mechanisms that right now we don't understand. And that's why I want to emphasize that research, basic research, is very important if you're looking for therapeutic strategies, because you need to know this basic science. So a few words on this extracellular tau. First, There was a number of reports in the past showing that tau was extracellular, it was toxic. But most of this report, especially those of Jesus, was unknown in the literature. I think that because everybody came out with this, for instance, this article by Amy Puller showing that there is secretion of tau, but it was known that tau was secreted and was also binding to some receptor, for instance, and have some interference with biology. So how can tau is secreted? It can be, like I told you, a normal secretion as a free forms, but it can be also in extracellular vesicles. Maybe you have heard about exosome, microparticles. So you can find tau within these vesicles. So I'm not going to go into detail here, but I will say a few words later on. Now let's move to tau pathology. And as I told you, of course, tau aggregate in Alzheimer's disease, but it also aggregate in other neurodegenerative disorders. So in Alzheimer's disease, you get the aggregation of a 6-tau isoform present in the brain, but in other tauopathies meaning these neurodegenerative disorders with tau aggregation, like Alzheimer's disease, Pick's disease, progressive supranuclear palsy, corticobasal degeneration, frontotemporal lobe degeneration, even some neuromuscular disorders. There's a number of, you see tau aggregation here, for instance, in progressive supranuclear palsy, in corticobasal degeneration, in argyrophilic Grand disease, and here in frontotemporal dementia in Pick's disease. And each time you can see that these aggregates are different. So we looked at the molecular level, what was going on, and in Alzheimer's disease, you have the 6-tau isoform, which aggregate into filament. But now if you look in progressive supranuclear palsy, for instance, you have only the 3-tau isoform with 4 microtubule binding domain. And in Pick's disease, you have the 3 isoform with 3 microtubule binding domains. And here it's an example of myotonic dystrophy. In this case, you have only the aggregation of 1 isoform. So of course, that's very puzzling. And we have to understand what's going on with this tau aggregation. So we have looked at the etiology of these tauopathies. And in tauopathies, you're going to have, for instance, dementia pugilistica, chronic traumatic encephalopathies or trauma brain injury. We're talking a lot about that, like rugby player, American football player, hockey player, and of course, boxer. And when you have a trauma like that, you have tau aggregation. If you look at children, for instance, with autistic children with self-injury behavior, maybe you see these kids knocking their head on the wall, they develop neurofibrillary degeneration also. There is also infectious, we're in Spain, the Spanish flu, with post-encephalitic Parkinsonism. And also measles can lead also to SSPE and, of course, tau aggregation. Metabolic disorder like Niemann-Pick's disease type C, and also toxin like on the Guam Island, the French Caribbean, or even in some cities where huge industrial waste. There is also some genetic effects, and I will emphasize a little on these genetic effects. Just to illustrate this diversity in etiology, I took the example of progressive supranuclear palsy because for any clinician, progressive supranuclear palsy is pretty clear. There's sometimes a mix with corticobasal degeneration, but it's a defined clinical entity. If you look at the neuropathology, it's not always true. And if you look at the time course of a disease, it's also complicated. I'm talking about that because it's a typical tauopathies. And some big pharma think that PSP can be a good model to test drugs for Alzheimer's disease because it's an orphan disease, so it costs less money to go in this kind of clinical trial and to go for Alzheimer's disease. But the process is very different. We will see that in Alzheimer's disease, neurofibrillary degeneration lasts for at least 20 years. So it means that you have a therapeutic window, which is pretty important. In PSP, it's a very, very quick disease, which leads to death very rapidly. So it's a big difference, and we see that the etiologies also are different. So if I'm taking the example of classical PSP, I mean, it's different from the ALS, amyotrophic lateral sclerosis, Parkinsonism, dementia complex of Guam, but it's very similar to the PSP which encountered in French Caribbeans. And in this case, we think that it's related to toxin present in fruits, and these toxins are, in fact, mitochondrial complex 1 inhibitors. So there's a number of articles showing the link between these mitochondrial complex inhibitors and the appearance of tau pathology. In the north of France, we linked also PSP to industrial waste, and especially when you have high increase in arsenic concentration, you will find an increased number of PSP patients. I'm sorry, I cannot go into detail for everything. The other part of PSP is related to genetic factors. So I'm coming back to the tau gene, MAPT, and which is located on the chromosome 17, and by alternative splicing, you raise the 6 tau isoforms. So the first thing identified in PSP was, in fact, that there was some genetic linkage with the H1, H2 haplotype, meaning that your gene is one way or the other way. Clearly, that's pretty simple. So there is an association signal in the tau genes for PSP and also for corticobasal degeneration. It's still pretty controversial. We show also that there is epigenomics changes. For instance, if you study the human brain in frontal cortex where you have tau pathology, or in occipital cortex where you don't have any pathology, you will see a decrease in methylation in PSP patient on the promoter of the tau gene. So there was a replication study showing you get this decrease in methylation, and you have an increase specifically in the expression of 4-repeat tau isoform in the frontal cortex, but not in the occipital cortex where you don't have any pathology. So epigenomics may be related to toxin or other stuff are also related to the tau pathology. As I told you, there is also an increase in 4-repeat tau isoform, meaning that you get 4 microtubule binding domain, as explained yesterday by Eckhart, and you have this typical profile with a upper tau doublet. And it means that you have more 4-repeat tau isoform than 3-repeat tau isoforms. So we look also if this splicing was involved in the story, and regarding always epigenomics, we were able to show that there is some microRNA decrease in PSP, which modulates splicing factors, which may explain the incorporation of this additional sequence in transcript and increase in 4-repeat tau isoforms. So, this idea of splicing, if you get aggregation of 4-repeat tau isoform, 3-repeat tau isoform, the 6 tau isoform, or only one without any additional sequence, is of course very interesting. It's very interesting because it may explain the differential aggregation of this tau isoform. And we know that there are some mutations on the tau gene. Yesterday, Eckhart talked about that. And these mutations are mostly located in the microtubule binding region and may explain a differential splicing, either increase in 3-repeat compared to 4-repeat, or increase in 4-repeat compared to 3-repeat. So these mutations are very useful to understand that factors that may lead to neurodegeneration. So we study a lot this mutation, not always involved in splicing, but there is a huge heterogeneity. I took two mutations, the one within the microtubule binding domains, and I mean the lady who had the founder mutation died at 60 years old, so it's pretty young, and the two sons have onset of the disease at 50. On another mutation, it's even worse, because the teenager was 17 years old, and the father did not show any symptoms, and he had the mutation. So you see also that there is huge differences in the onset of the disease for the same mutation within the same family. So it's very complicated, because when you have a young teenager, depressed, not interested by anything, you're not thinking about neurodegenerative disorders. So you don't see a neurologist, you see a psychologist. But she started at 17, she died at 24. So you see, we have to be very cautious now in following this patient. So the idea is that when you change any tau isoforms within the neurons, it may lead to aggregation. So is it related really to splicing, or is it related to subneuronal population? In fact, it's both, so it may be more complicated than what we thought. Anyway, splicing is important, and for that we use a disease model, which is myotonic dystrophy. I don't know if you know myotonic dystrophy. It's like Huntington's disease or spinocerebellar ataxia. You have a poly-CUG track increase. I say CUG because, I mean, it's never translated into protein. So it means that we have transcriptional CUG and CUG tracks, and these CUG tracks stay at the mRNA level and are able to sequester any splicing factors. So the idea is that if you get splicing modification, you will have disease. And myotonic dystrophy is wonderful, in a way, for studying splicing, because these patients have diabetes, but it's related to a defect in splicing in the insulin receptor. They have myotonia. It's related to defects in channel chloride, in splicing in channel chloride. They have heart problem. It's related to splicing in troponin T gene. And they have muscle weakness, and we don't know exactly which of the gene is responsible for this muscle weakness, but all of these genes have splicing problems. And finally, they have cognitive deficit, and there are some splicing I show you in tau, but also in other genes like NMDA receptor or APP. So to make a story short, I mean, really we have shown that there is a link between the splicing of tau and the cognitive defect, and that in fact it's related to a family of splicing factors called muscle blind. And recently we shown that muscle blind is really the key family of proteins involved in myotonic dystrophy for aberrant splicing in most of these genes. Okay, coming back to tau now. A few things on background on tau models. I mean, now we know we have 6 tau isoforms in the brain that they aggregate and lead to neurofibrillary degeneration. Yesterday, Eckhart showed you this kind of picture saying that it's one of the hypotheses that if you have hyperphosphorylated tau, you get depolymerization of the microtubule and axonal damage. I mean, for most of us, I mean, we are not sure it's really like that. Why? Because you don't have a huge loss of white matter in Alzheimer's patient, for instance. So meaning your axonal loss maybe, yeah, axonal defects, but not really axonal degeneration. The other thing I was telling you is that, in fact, it takes a long, long time for neurons to die with neurofibrillary degeneration. Globally, I mean, we have like a 15 to 20 years time before to go from appearance of tau aggregates until ghost tangles, meaning really a ghost of aggregated tau. So how to study this neurofibrillary degeneration? Yesterday we told you that if you try to overexpress tau, you don't have any aggregation. So the best way is to use this mutation I told you about, and Eckhart told you. There is some pro-aggregating mutation, but there is also other mutation. You can use them in worm. Yesterday we have a quick view on worm and how you have degradation. You can use that in fly, in fish, in rodents, and even in non-human primates. I will focus, of course, on rodents here. The other thing we have to say is that we have really a therapeutic window. Yesterday, I mean, it was a clear demonstration. When you switch off the transgene for tau, pathological tau, you rescue really the phenotype. There's a number of papers like that. There's also papers showing that when you start neurofibrillary degeneration, I mean, if your tangles is within neural network, your neural network is still functional. So in the lab, we use a specific model with 2 mutations. Why 2? Because, you know, we're French. We always think that 2 is better than 1. Honestly, I'm not sure that it did anything different than 1, but when it's done, it's done. Anyway, and we were lucky enough to have expression of this tau transgene only in the subiculum and the CA1 layer of the hippocampus. So it's really the region which is affected in Alzheimer's disease. So you have aggregation, and we have a nice correlation between the appearance of neurofibrillary degeneration and cognitive tasks, like here, cognitive deficit in the Morris water maze. I'm not going into detail, you have to trust me on that. We have also some synaptic defects, either by LTD and what we call the BDNF synaptic facilitation, so BDNF LTP. We don't have anything in classical LTP. So there are some synaptic defects in these mice. Can we use this kind of tau transgenic mice to study modulating factors and to see how our daily life, or can we have therapeutic strategies using these models? So we have to come back to Alzheimer's disease and epidemiology. So we know with a genome-wide association study a number of genes involved for the risk of Alzheimer. But we have also a number of environmental factors banked yesterday or the day before. No, yesterday. Yeah, time lasts for very quick in Alicante. Banks show that you have multimodal studies where you're going to do physical exercise, you're going to change your diet, you're going to change many things, and you see improvement on cognition. So we say, okay, can we look at environmental factor? We looked also at genes, but I'm not going to focus on that, maybe a few things, and see if we can modulate the daily life of our tau transgenic mice. If I'm talking about daily life, it's always in reflect also to what's going on from GWAS, and in GWAS we have seen that cholesterol pathway is very important, the immune response is very important, endocytosis, I mean, I talked about tau secretion, endocytosis of APP and amyloid formation, so it's pretty evident, and cytoskeleton also. And you see, of course, APP and tau. Anyway, but we get interested in cholesterol immune response and endocytosis a little. But regarding cholesterol and immune response, it's interesting because that's something we can modulate. So Which factors influence Alzheimer's disease risk? Environmental factors. It can be physical exercise or coffee. We're just finishing the coffee break. So we had a prize on resveratrol. So resveratrol is part of red wine, okay? Anesthetic agent and other stuff. I will not develop on that. What we have looked is that high fat diet, so obesity, will increase tau pathology. If you're doing physical exercise, you're going to decrease tau pathology. Now, if you get anesthesia, and that's pretty terrible because anesthesia, you get like this post-operative cognitive decline, and most of the patients are fine after a couple of hours. But think about elderly people. They have usually repetitive anesthesia. So when we looked at anesthesia, you can see that there is a reversal of tau modification leading to aggregation. But if you do repetitive anesthesia, you don't have this reversal anymore. So it means that it's not anesthesia as a single anesthesia, which is a risk factor. It's the repetition of anesthesia, which is a risk factor for cognitive defects in mice. No, but I mean, we have to be careful. I mean, you know, I can finish like that and say, okay, and then what happened in human? I mean, who want to get a hip replacement without anesthesia? Nobody. Okay, if you look at physical exercise, for instance, we looked at these omics approaches. So you can do transcriptomics, proteomics, any omics you want. You can see things move, not a lot. If you look at cholesterol pathway, the only thing which really moved are CYP46 and a little NPC1 and NPC2, but I'm not going to talk about that. CYP46 is an enzyme involved in cholesterol esterification. And esterification of cholesterol is very important because it's how it's getting to your brain or outside of the brain. So if you have esterified cholesterol, it will be very important. If now you use gene therapy approach and you inject CYP46 in our mice, you can rescue everything in our mice. Behavior, synaptic plasticity, only by modulating cholesterol. Even neuroinflammation is decreasing following that. Coming back to that, okay, so we have a link to cholesterol. If you do the same thing with this enzyme in an amyloid model, you get the same data. So meaning that cholesterol brain homeostasis is very important. We looked also at neuroinflammation because everybody say, okay, in Alzheimer's disease we have two brain lesions, amyloid deposits, and neurofibrillary tangles. But everybody say also, and we have a huge neuroinflammation. Yeah, that's not multiple sclerosis, so yeah, you have inflammation, it means you have gliosis. So what can we do with that? We were interested to this inflammation because, in fact, when we did the first transcriptomic studies in our mice, the first family of genes coming out was related to inflammation and the immune system. So we say, okay, there is something really important here. And we just finalized these two studies showing that, in fact, yes, there is a huge increase in a family of genes involved in inflammation. We have microglial activation, chemokine production. We know that chemokine, especially the one we study, which is CCL3, interfere with synaptic plasticity and may explain cognitive deficit. And we have also T-cell infiltration. And we were puzzled by this T-cell infiltration, but we looked at this FTDP17 mutation, you know, the tau mutation in the microtubule binding domains. And in this patient, we see also T-cell infiltration. So we say, okay, if we get CD8 positive T cells in the brain, where it's coming from, and to make a story short, we say, okay, we have to remove these T cells. So what we use is an anti-CD3 immunotherapy, something used, for instance, in model of multiple sclerosis, but this time we did an anti-CD3 immunotherapy for a long time, from 3 to 9 months of age, and at the end we were able to stop T cell infiltration. Of course, we don't have any T cell anymore, and we were able to have improvement in spatial memory, meaning that this stopping the T cell infiltration allow for decreased chemokine production and allows also to restore cognitive behavior. So we have now a link that T cell infiltration is instrumental into the cognitive impairments promoted by tau pathology. So can we modulate this neuroinflammation? We thought about coffee, because coffee is an antioxidant and is known also to play on oxidative stress and inflammation. I mean, epidemiological studies say, okay, caffeine is good also for the amyloid. So we say, okay, caffeine may be a good stuff. And we were able to show that, indeed, caffeine is able to decrease tau pathology, decrease inflammation in these mice. So it means that with these mice, you're able to study different environmental factors. Okay, for those who works on caffeine and knows caffeine, you know that caffeines have many targets. And one of the targets is also an antagonist of A2A receptor. So we checked that also, and we show that, in fact, when you breed the mice, the tau transgenic mice, with mice knockout for the A2A receptor, you also rescue the phenotype of our tau transgenic mice, meaning that similarly, if you use A2A receptor antagonist, it's good for the tau pathology. You rescue cognitive deficit and decrease neuroinflammation. I'm not going to the detail, but that's the idea. So with everything now, we have a number of therapeutic approaches. That's number of therapeutic strategies. I think that Bengt already talked about most of them. So I'm not going to go into details. Just to say that you can do this kind of work in these tau transgenic mice. You can test active immunization, passive immunization. Here it's an example of active immunization. Here it's the Roche paper showing that you can use also active passive immunization with an antibody against phosphoserine 422. I should say that the clinical trial stopped in phase 1 for Roche. So even if it works in mice, it doesn't mean that it will work in human. It's very important to have this in mind, because if you're thinking about tau, if you inject tau, human tau, to normal mice, you get an autoimmune response. So the idea to have phosphoserine 422 is that this is a kind of pathological epitope, which is not present usually in normal mice. So even with this approach, they show that you have risk in human. So on the first conclusion, I will say, or maybe final conclusion, do we still have time? 3 minutes, okay. I should say that tau proteins are more than MAP. There is a development of different animal models allowing for new therapeutic strategies, but we have to think of how good are these models, because it's always overexpression. Talking about tau immunotherapy, okay, you decrease tau levels, but you overexpress tau so much, what does it mean? So people are going now, really, they are knock-in models to trying to have pathology using the same level of protein. Very quick, I will talk about the BRAC stages, what we have seen in Alzheimer's disease. It's true also in progressive supranuclear palsy, in argyrophilic Grandt disease. Many people have shown that now that tau is able to be transferred from cell to cell, and maybe through transsynaptic mechanisms in vitro. And I told you already, it can be in 3 forms, aggregated or not. using extracellular vesicles like microparticles, exosome, or tunneling nanotubes. I'm not going to go into details, but we and others have worked a lot on that. There is also in vivo model of this propagation. We developed a rat model on that, showing that you can have a transfer of the tau pathology. And the main problem now is if you have a transfer of the tau pathology, is it a prion-like mechanisms? Meaning when you start to have a kind of aggregation, if you inject these aggregates into mice, you have pathology. But I mean, it's, you know, yesterday Eckhart showed you this kind of picture that monomer of tau can go into protofibrils, but the first step here are very difficult in a thermodynamic way. So to go faster, to go faster on that, you can add seeds, meaning you can add brain extracts, and speed up the nucleation process, meaning you're going very fast this way, and then for the elongation, it's a quick event. So if you're able to lower the number of the time to aggregate tau, you can increase pathology. I can show you that here. If you inject Alzheimer homogenate or Alzheimer fibers, you are able to develop pathology in mice here very quickly. Why I'm showing that? Because it's very useful to test also antibody. Here we test different antibodies. You can see that this antibody will decrease the seeding of these tau aggregates. So meaning it interferes with tau aggregation. It doesn't mean that we have tau strains, meaning that we have specific tau aggregates which may explain only the aggregation, for instance, of 3-repeat tau isoform, 4-repeat tau isoforms, but it means that you can speed up the process. So if your tau aggregates are transferred from one cell to another, healthy cells, you can imagine that these healthy cells can develop pathology. So that's why on the social networks you're able to see that you have, I don't know, a kid saying to his mother, why I'm not able to visit grandpa? And the mother say, grandpa has Alzheimer's disease, is contagious. Okay, no, it's not contagious, okay? So, just to thank the sponsor and all my team, and sorry for being a little over time.
Thank you very much. We have time for one or two very short questions.
I found very interesting the topic about anesthesia and what happens when it's repeated anesthesia. And if you like, could you comment about the possible relation between anesthesia and hibernation? Because I wonder if a repeated hibernation that takes place in some mammals could also be similar or could also to induce some kind of tauopathy or not.
Well, anesthesia is complicated because when you're talking about anesthesia, you have to talk also about body temperature. So, because when you get anesthesia, usually your body temperature drops down. And if your body temperature drops down, you get hyperphosphorylation of tau, and you get modification in the biology of tau. But if you do everything in normothermia, you control body temperature, you still have this modification of tau, and you still have deficit. Yesterday, Eckhart told you phosphorylation is not a requisite for aggregation. And I think he's right. Now, I will say hyperphosphorylation of tau is linked to cognitive deficit without aggregation. Because if you get hyperphosphorylation and chronic hyperphosphorylation of tau, you get cognitive deficit. Following our work in anesthesiology, a Korean group did the same experiment in knockout mice. They didn't see any change in cognition, meaning that, in fact, the loss of, the cognitive deficit is really related to the tau hyperphosphorylation. So, then after there is other things about torpor, hibernation, where you have also tau hyperphosphorylation, and it can be also, I don't know, if we're talking a lot about these tau strands, this signature of tau species, it can be a kind of memory for the cell. If you get aggregation in bears or in Syrian hamster for tau, it can be kind of memory of the cells where exactly after the hibernation coming back, they have a kind of memory card with this specific pattern of phosphorylation. That's a kind of crazy idea we can have.
Look, thanks for a good talk. I had a clinical comment on this, that we saw that elderly people after anesthesia came into some confusional state often, and perhaps even a dementia condition. But if you correct really the per-operative anesthesia, looking on not getting hypoxia, not getting blood pressure falls, then we really could almost exclude confusional states. So I think oxygenation is a part of it, and lowering of blood pressure. So you have to teach your anesthesia nurses how to really supervise elderly patients.
Yeah, I fully agree. I mean, I was talking about body temperature, but we have a number of factors which need to be controlled. But we have also to be very cautious when we have patients coming for anesthesia or any surgery to know exactly also their, let's say, cognitive status, because it's still a risk factor if there is a huge number of anesthetic intervention. I mean, intervention with anesthesia.
I also had a short question. Have you really seen T cells in the human post-mortem Alzheimer brain?
Yes.
Because if I look, it's glial cells, it's astrocytes, but T cells is for me.
Yeah, we've checked that with people who used to work on T cells in Parkinson's disease, and they looked in frontotemporal dementia with tau mutation similar to the one we use in our animal model, and they are sure they are T cells. So different techniques, and so we are sure for T cell infiltration in this case. Honestly, it's not a huge T cell infiltration. It's not multiple sclerosis. So.
All right, so I think we have to move on. Thank you very much.